The respiratory system. We want to cover the different organs of the respiratory system through the conducting portion and the respiratory portion of the respiratory system. We want to follow the path that the air goes through from the outside, interacting with the environment, to the inside of the lungs. And we want to identify the cells and characteristics of cells and function of cells that line the air spaces, not only the conducting portion, but also the respiratory portion where the gases exchange occur. We'll end by talking about some disease states of the respiratory system. Here we see some lungs, which is the main organ of the respiratory system, but we'll see there's several different organs involved. What we want to do today is to characterize the subdivisions of the respiratory system, identify each cell type and the matrix involved, describe the structure and function of the different components, including the macrophage, type 1 and 2, alveolar cells. We also want to identify the passageway found in the lungs, alveolar ducts, alveoli, bronchioles. Now, the respiratory system has two portions. It has a conduction portion, or conducting portion, which starts with the nose and mouth and goes all the way down through a series of branching in the lungs down to the alveolar ducts, and here, and so the function is to carry air down into there, and you have open passageway to the environment itself. Also, the conducting portion does three important things. It warms the air, and we'll talk about how the blood flow facilitates that. It moistens the air, and so we will expect to see some glands and that secrete fluid in through here. We also expect to see goblet cells along the way, and it filters the air. And so there's hairs in the nose in through there, and there's different ways in the conchi to filter the air before it gets to the lung. So this is all the conducting portion way down here to the respiratory bronchioles. And then we get to the respiratory portion, and the function is to exchange gases with the environment, oxygen in and carbon dioxide out. Also, this area produces surfactant, which reduces surface tension and prevents the closed alveoli to fail to open. And their structure components, we have cartilage, a couple of types of cartilage. We have laminar propria connective tissue, and a very important thing is the vascularization that we will see. Now, there's different types of epithelium. Remember, when we talked about epithelium, we had stratified squamous epithelium, flat ones at the top, but you have multiple layers. In contrast to the simple squamous, which has just one layer of flattened. And then pseudostratified columnar, which is really what we call respiratory epithelium. It's really misnomer because it's in the respiratory system, okay, but it is not involved in the respiration itself. It's actually these cells that does the gases exchange to occur. And so we have stratified squamous epithelium, respiratory epithelium with ciliated cells and goblet cells, and then you have simple squamous epithelium, which actually touches the air space. In addition to that, there's olfactory epithelium, which is really kind of like this type of epithelium. It does have ciliated cells on the surface, but you have multiple cell layers in the olfactory epithelium, as we'll talk about a little bit later on. And if you look at the different portions of the airway, you can see the different types of epithelium. The first portion is interacting with the skin. So at a skin junction there, you have stratified squamous like you have covering all your skin of the epidermis. If you come on down into the nasal cavity, and that's what we see here, we pick up respiratory epithelium with ciliated cells and goblet cells, and that persists around the false vocal cords, and then it continues after the true vocal cords, down into the trachea, all the way down into the bronchioles. That we'll see respiratory epithelium of different heights. And then when you get to the very at the alveoli, you will see simple squamous epithelium. So the different types of epithelium that we'll see. The stratified squamous not only is at the opening of your nose, but it also covers the true vocal cords that we see there. So there are different types of epithelium, and it's found at different locations. Now this is one slide that we don't have. It shows you true vocal cords and the false vocal cords, but the slide that we had, 38B, shows you the false vocal cords. There's some lymphoidal tissue associated with that, and the true vocal cords, we have the muscle, the ligament, and the stratified squamous epithelium on the surface, and we have the cartilage, the same kind of cartilage, the thyroid cartilage, and then the tracheal cartilage that we see over here. Now, respiratory epithelium is pseudostratified, that is, all the cells touch the base. There's the basal cells, there's goblet cells, and there's ciliated cells. You can see the basal bodies and the cilia projecting there. Scanning electron microscopic view shows you the mucus-secreting goblet cells, and then you can also see the cilia projecting into the airspace to move the fluid along. So another view of that, these are the goblet cells that secrete mucus, and you can see the mucus being projected from those. You have the ciliated cells. There's also basal cells in through there as well. And what happens is the goblet cells secrete a mucus line the airspace, so the air is going through there. Dust particles get trapped in there, and then the ciliated cells, with their actions, move the dust along like that little boat, just washes it away. But where does it go? It's about a Coca-Cola can full a day, and so you swallow that. And so these are the cells that are destroyed during the smoking process, and so you don't have a natural way of getting mucus out of the smoker, and so they have to cough it out. So they have two options: to drown in their mucus or to cough it out, so they cough it out. But normally in normal tissue, ciliated cells move that mucus out about a Coca-Cola can full a day. And here we see the respiratory epithelium on the surface with the goblet cells, the basal cells, and the ciliated cells. And below that you see a very thick basement membrane, very characteristic of respiratory epithelium. It has a very thick basement membrane. And then you'll note the vascularization. There's lots of blood vessels in through there. And then there's some serous glands, as we will see. Now, the highly vascularization, as you can see, into right underneath the respiratory epithelium, is important. This is a lamina propria. This is cartilage in through there, one of the cartilage components, and it's important in conditioning the inhaled air. A complex vasculature loops, these loops in here, go in a direction from which the air is coming. So air is going in this way, and the blood is going that way, and that warms the air, which is very important. Lungs is not like cold air. If you've ever been jogging on a real cold day, I feel your coolness in lungs doesn't like cold air, and you can actually feel that in the coolness of your lungs on a cold day if you're jogging and breathing hard. Also, these rich vasculature is associated with swell bodies. Swell bodies reduce the flow of air on one side of the nose or the other side, and it switches back and forth every 30 minutes. And that will allow rehydration of one side of the nose and the nasal cavity as the air is flowing down through the other side. It'll switch about every 30 minutes. So here we see the respiratory epithelium, ciliated cells, goblet cells, basal cells, and the same as what we see there. Here, respiratory epithelium, as we'll see here in a minute, but a rich vascular supply in through there, and the swell bodies, these are the swell bodies that whenever they swell, they close the airflow into that space. In your nose, you have the hyaline cartilage as well. There's Bowman's glands in here to discharge onto the surface to help humidify the air. And pseudostratified columnar respiratory epithelium is what we see lining that. So these are the different turbinates, and the one at the very top is the one that has olfactory epithelium that allows you to smell. And here we can see olfactory epithelium has many cell layers, and also it has ciliated cells on the surface, but it's not the same type cell. You can see the cilia there are on actually nerve cells that goes through there. So these are really receptor cells, nerve cells, that have the cilia, very long cilia, that are in the airspace for it to detect it. And then you have all these supporting cells, which is a high epithelium that we see. So here we see, right in this region, you have the respiratory epithelium in this region, but in through there you have olfactory epithelium. There's Bowman's glands in through there. These are a couple of artifacts right in through there, and there that is bone in through there. And here we can see a higher magnificat. This is a multiple cells of the respiratory epithelium, and you can see the cilium of the receptor cells, Bowman's glands, and also note these rich concentration of nerves in through there. There's a perineurium. Endoneurium would be on the inside in through the rich vascular supply, and these glands to humidify the surface. Here's the olfactory epithelium and the regular respiratory epithelium, the swell bodies, and Bowman's glands in through there. Now here we see the larynx. Here's the tongue. This is the epiglottis, and these are different cartilage. This is a thyroid cartilage, as we saw here, a conchoid cartilage that we see there, and a tracheal cartilage that we see. So these are hyaline cartilage. However, the epiglottis itself has elastic cartilage, so it can be pliable. We can see the epiglottis in through there with these are actually artifacts in through there. If you go to a higher mag, you can actually see the fibers, the elastic fibers that are in the cartilage there. On one side, you have non-keratinized and stratified squamous epithelium, and then that's the one that touches the oral cavity. And then on the other side of it, you have the respiratory epithelium. Remember, respiratory epithelium is pseudostratified columnar epithelium, as opposed to a stratified squamous epithelium, but it's non-keratinized, as we can see. That's elastic cartilage that we see. Now, on the superior surface, you have non-keratinized stratified squamous epithelium. When the epiglottis is closing the larynx, this layer is exposed and acts as a barrier to prevent swallowing food from getting into entering the larynx. The inferior one is the respiratory epithelium. This layer is exposed during the breathing. So one is exposed when you swallow, and one is exposed whenever you are breathing, and that's the two different types, either stratified squamous on the swallowing side, or the respiratory on the non-swallowing side. And here we see a larynx. This is a true vocal cord, false vocal cord in through there. This is a cartilage, the thyroid cartilage, and this is tracheal cartilage in through there. There's two types of cartilage found in the respiratory system. Elastic cartilage only occurs in the epiglottis. All the other ones are hyaline cartilage. So these are hyaline cartilage in here, and the elastic cartilage that we just saw is in the epiglottis. Now this is a slide that we will see to look at the false vocal cords right in through there, and the true vocal cords in through there. There's the muscle, the skeletal muscle that we could control the pitch of our sound, the ligament associated with that. And then there is stratified squamous epithelium, which is undergone metaplasia or transform from the respiratory to the protective type of epithelium, and more of this would transform to this protective stratified squamous epithelium if you were a smoker. So here we can see the glands in the vocal cord, stratified squamous epithelium, the hyaline cartilage. You can see the muscle, the skeletal muscle, the vocal ligament of the true vocal cord, the respiratory epithelium, the false vocal cord, true vocal cord, as we can see there, and the true vocal cords has the non-keratinized stratified squamous epithelium. As you come on down to a lower portion, we can see the thyroid gland. This is the follicles of the thyroid gland, skeletal muscle, and through here, hyaline cartilage. This is hyaline cartilage. Connective tissue is all throughout there. It connects everything. Respiratory epithelium is what lines the lumen of the trachea, and then we can see the esophagus, and there's brown fat. This is brown fat right in through there. You can see glands, respiratory epithelium, cartilage. This is a lymph nodule that we can see. So here we see the trachea again. In humans, there's 16 to 20 C-shaped rings, so it doesn't make a whole circle, it just makes a C shape. And there's a muscle right in here, smooth muscle that's associated with that. As smooth muscles allow for regulation of the size of the lumen. So when you cough, you want to increase your velocity of air, and you squeeze that down. Is what happens. And here we see in the monkey, this is a smooth muscle there. There's glands. This is we're talking about the trachea here, not the esophagus. This is part of the esophagus here. Hyaline cartilage, perichondrium. This is lamina propria, and maybe you might call this submucosa by that time, but there's glands that are located in through there. Now this muscle allows you to narrow the lumen. The capacity to narrow the lumen of the trachea by contraction of this muscle is important in the cough reflex. Narrowing the tracheal lumen increases the velocity of the expelled air and better loosens the material, so you're able to blow out the butter beans that got down to the wrong place, for example. Coming down from the larynx, we have the trachea, and you have a series of branching and branching and branching in different types. When you first branch, it's a bronchus, and then you get to bronchioles, and there's different lobes in the lungs, and you have these cartilage rings, which are, this is hyaline cartilage. Now, if you would take caulking, like you see it's a caulking gun, and put it inside the lungs, it will take the shape of the air spaces, and here you can see well, air goes through bigger pipes and smaller pipes and smaller pipes. All this is a conducting portion of the respiratory system. And then at the very end, the little grapes down there are the alveoli that's associated with that. So here you can see this is a horse's lung and one of those casts of the air spaces. So you have a series of different shapes, branching and branching and branching in the lungs. All this branching increases the surface area, and in fact the surface area is about the size of a tennis court. So in humans with both lungs, the air exchange that is between this portion and the blood vessel inside there is the size of a tennis court due to all the branching and branching and branching that we have. And here we can see the trachea, the cartilage, the respiratory epithelium, the lamina propria, glands in the submucosa, and here we can see the muscle that we have there. This is the trachealis muscles with that when air contracts, it closes the C and increases the velocity of airflow. Goblet cells and ciliated cells that we see there in this pseudostratified columnar epithelium. Nerve, there's the perineurium right there, endoneurium around each cell, blood vessels, lymph nodules that we can see there. The trachea, there's 16 to 20 C rings, as I mentioned, and there's smooth muscle that facilitates that. Here we see the trachea, and then it branches into the two bronchi or bronchus, as you can see there. So that branches. So now this is in the right or left lung. So here we see the bronchial tree, and there's various types of bronchi. You got the primary, secondary, tertiary bronchi. As it goes, it gets smaller and smaller, as you can see. And finally, you get to the tertiary bronchi, small bronchi, and from there you get to the bronchioles. And it has a series of bronchioles. And all this branching and branching and branching is really the conducting portion of the respiratory system, even within the lungs itself. Finally, you get to terminal bronchioles, and then from there you get a respiratory bronchiole that have little outpocketings. For these, we can see that these outpocketings here are the respiratory bronchioles, alveolar sacs, alveolar ducts that we see through there, as we can see, but this would be the respiratory bronchiole going through there, terminal respiratory, and each one of these little ones is an alveolus. And here we see the cells of the respiratory portion, because we're out of the conducting portion into the respiratory portion. We have the terminal bronchiole, that's the last one that doesn't have exchange to occur. And there are certain cells in here. There's Clara cells. I'm not sure what they do, but they're in there. And there's also still, we've got ciliated cells, but the height may be reducing. There's also elastic tissue, and you can see the elastin, the black elastin that's around through there, because the lungs will contract as part of the breathing process. And there's also smooth muscle. So we can see the pink smooth muscle here and here and here. And here we can see smooth muscle in this toluidin blue specimen in through there. As you get down to the alveolus, you see the type 1 pneumocytes. That's the one who touches the air. That's the one that has the gases exchange. We also see the type 2 pneumocyte. Those are the ones that produce surfactant. In the lumen, we can see dust cells or lung macrophages. Here we can see the macrophages, another one right there. Also, we can see the endothelial cells that line the capillaries. And so, in this little space that we have right here, we can see it right there very nicely. You have endothelial cell on the inside and a type 1 pneumocyte on the outside. So in the lung, we have the bronchus, and we know it's a bronchus because it still has cartilage. When it no longer has cartilage, either a cartilage ring, like in a trachea, or these plates, it no longer has that. It becomes a bronchiole, conducting bronchiole or terminal bronchiole. You have a respiratory bronchiole that has some outpocketings that is exchanging air. Alveolar duct that we see here. Actually, this looks more like a respiratory bronchiole with the alveolar duct at this end. Alveolar sac, which is just one of these projections is a sac, and then the alveolus. As we can see, there's a blood vessel right there with some neutrophil in through there. These are also blood vessels in the wall, as we see there. So in the conducting portion, you come through a series of branching and branching and branching. And not only is there branching of the airway, but there's also branching of the blood vessels as well that parallel this branching as we go through there. And you can see that around each alveolus is a network of capillaries. In fact, capillaries was discovered in the lungs. For a long time, they knew about the arteries and veins, but they didn't know where they went back together. And then they discovered capillaries in the lungs because the air spaces didn't impede them viewing those. So this is the blood-air barrier. This is air, blood, and you can see that there's 3 components to it. You have the type 1 pneumocyte, which has a gaseous exchange, and then you have the endothelial cell lining this blood vessel, and then you have a fused basal lamina. basal lamina, these two cells together. So those are the three components of the blood air barrier. And here you can see it in the slide 42. This is the red blood cell. This is air right through there. This is how thin it is in relation to the eight or so microns red blood cell. You can see this is much thinner than that. And so here we can see this is what we're seeing here is a capillary network that's is in the wall of the alveolus. So you got the respiratory bronchiole coming in here, alveolar duct, alveolar sacs, and then an individual alveolus. Also, there's little pores in there, little pores that allows the air to equalize, as we can see there. Blood vessels, ciliated cells continue, but when you get the alveolar duct, they're not ciliated anymore. They're just cells there without the cilium. And also you don't have goblet cells down in this region. So before the respiratory bronchioles, you stop your goblet cells. Don't have goblet cells in this region because that would interfere with gas exchange. There are other cells in the connective tissue, and here we see a mast cell. Here, 48 slide, we see a mast cell. Here is a light staining euchromatic nucleus, and we can see the granules. Mast cell functions in local release of many bioactive substances associated with the immune response, tissue repair. So mast cells, granules, those granules contain heparin, histamine, proteases, eosinophil, neutrophil factors, as well as cytokines. So mast cells interact with these other cells by stimulating chemicals that react to them. In addition to having mast cells there, we got smooth muscle in the wall regulating the conducting portion of the respiratory system. We see nerves that would be important in regulating these muscles. This is a myelin axon, the perineurium, fat cells, and in this particular space, the blood vessel itself from the heart has cardiac muscle. Sometimes you can have cardiac muscle. Normally it's smooth muscle, right? But you can have some cardiac muscle in the wall of the major arteries going into the lungs. In addition to the different cells that we have actually lining the space, we also have some cells in the middle. Those are the macrophages, the air macrophages, which gobbles up whatever may be going in through there. So we see like the terminal bronchiole and then the respiratory bronchiole. This is probably terminal on this side, alveolar duct on that side, and then here we see the type 1 pneumocyte in through there, which has a gaseous exchange to occur. Also, we can see macrophages and other preparations. Here you can see the nucleus of the macrophage, the different things that it has taken up. As you can see, another macrophage in the wall there. And here they've taken up a lot of black stuff. So these are macrophages which gobble up the materials that is not filtered out during the process. So you have the type 1 pneumocyte, gaseous exchange, and type 2 is the one that produces surfactant. And the type 2 pneumocyte produces surfactant, which helps prevent alveolar from collapsing, and it reduces the surface tension to allow it to reinflate once it has collapsed, and it ease breathing because you don't have to pull it apart. It kind of falls apart. If you look at the outside of a lung, you see the mesothelium. So this is actually the visceral layer of the mesothelium, and this would be the pleural cavity right in through there, but it's been pulled, the lungs pulled out of it. Connective tissue, type 1 pneumocyte there, type 2 with this flocculent stuff, we can see it in through here and here and here. And then we have macrophages in through there, fibroblasts, and this is mesothelium again in through here, endothelial cells lining these red blood cells in the wall of these. And here we can see the type 1 pneumocytes here, here, here. A type 2 pneumocytes with a surfactant usually kind of in the corner, but you can see the surfactant located in there, and then we have macrophages there. So if we look at this slide, we can see some mast cells right in through there again, 42. But this is a bronchus. You can see the cartilage plates that will be there, conducting bronchiole, respiratory bronchiole, and the alveolar duct, alveolar sac. Mesothelium, that's a very good indication of mesothelium there. Alveoli, which is more than one, alveolus is one. Macrophages again, the type 1 pneumocyte, type 2 pneumocytes in through there producing their surfactant. And so we can see the macrophages in through there. Macrophages phagocytize red blood cells lost to damage the capillaries and the airborne particles. They phagocytize airborne particles. In congestive heart failure, the lungs become congested with blood, and red blood cells pass into the alveoli, where they are phagocytized by these alveolar macrophages. In such cases, these macrophages are called heart failure cells because they are phagocytizing cells as a result of the heart failure. Here we see a macrophage with the organelles. They got Golgi apparatus, machinery, lysosomes, mitochondria, ready to gobble up anything that's exposed to it. And this is from a smoker. A smoker already has its cells filled of debris from the tobacco smoke. And so they're less resistant than would be a non-smoker if they're exposed to environmental conditions. Here we see a little piece of lung from a human, normal lung, and this is from a smoker, as you can see there. This is from a smoker. This is all tar. Also, this one has emphysema. The word emphysema means too much air in the lungs, and you can see how the air is bulged out and through the air. And this one is another smoker, and this is a tumor in through there. And here we can see the surfactant laying over. There's the two EMs that we're supposed to see. Lining the type 1 pneumocyte is a surfactant. And here we can see the cells, a type 1 pneumocyte, where they kind of glued together to one another in through there with a type of epithelial junction, so it's on your occlusions, it's on your adherents. And so the type 1 pneumocyte covers about 95% of the alveolar space, about 95%. Some of the other of it would be covered, of course, by the type 2 pneumocyte. And we can see the occluding junction between adjacent cells, holding these cells together and preventing air from leaving or fluid from coming in. This is a type 2 cell. A type 2 cell, you can see the lamellar structures there, which is really their surfactant itself. So surfactant comes here and coats and covers the surface to reduce the surface tension. Here you can see the surfactant is like a lipid, a wiry lipid that lays over the surface. So surfactant function reduces the surface tension and reduces the work of breathing, and it helps keep alveoli open and may have a bactericidal effect. Hyaline membrane disease, immature infants cannot get or make sufficient surfactant, and so it's hard for them to breathe in the beginning because their lungs collapse and the little alveoli doesn't open up again because they don't have surfactant to reduce that surface tension. And so also there's fluid in through there. That's why you always have the macrophages, the ciliated cells to pump it out. And so that ciliary action is what removes it. Also, this has lysosome, collagenase, and different things to help break down proteins and things that are in there. And then you have macrophages. Macrophages contain hemosiderin, phagocytide lysis enzymes inside the fluid. And our clinical correlation, it had to do with smoking, where you had metaplasia, transformation of the respiratory epithelium, just stratified squamous epithelium that we see here. So in the air barrier, as I mentioned to you before, you have the fusion of the plasma membrane of those two. And then we have, in review, we have alveoli at the very end of a series of branching and branching and branching. And this is the respiratory portion, and then the conducting portion goes from here all the way up to the nose. And you have the mesothelium on either side, the visceral leaf, as we saw before, and parietal leaf, which we did not have. Now, in terms of diseases, this is a normal lung. Look at the amount of pink versus white. Pneumonia, there's too much pink. The walls are swollen. These black things are actually white blood cells that are in the space that should not be there. And this is emphysema. Emphysema is a loss of the wall. And as I mentioned, the word means too much air in the lungs, and they can expand beyond because you don't have these. If you stop smoking, the stratified squamous will transform back to the respiratory epithelium, and you'll lose some of that cough. However, if smoking induces emphysema, this will not reheal. Other respiratory diseases, you have influenza, bronchitis, cold, pneumonia, many things. Emphysema is one of those that doesn't repair, and it's because of loss of the wall that occurs. The end.